Today we're talking about Chapter 8, Section 5, which is the topic of translation. Now, before we get started here, you want to make sure you have the corresponding Power Notes sheet. This is what it looks like, and this one is a little bit different. It has a concept map, so you might need to watch the video before filling in the Power Notes just to get an idea of the overall process of translation to be able to figure out what goes in those boxes here. Then, of course, the diagram to label some steps here, and on the second page, we have the section quiz from your textbook. So once you have that ready to go, you can go ahead and watch the rest of the video and learn about the process of translation. Translation will occur in the cytoplasm of the cell, and it's going to require the use of all three types of RNA. Let's make sure that we review those types of RNA so that you remember what they are. The first one is going to be messenger RNA, or mRNA. Remember, the M stands for messenger. That is the message or the instructions from ultimately the DNA inside the nucleus that was used to make the mRNA during the process of transcription that happened before translation began. The next type of RNA used in translation is going to be the rRNA, or the ribosomal RNA. That is what ribosomes are made out of. So the actual stuff that is constructing the structure of that ribosome is rRNA. The rRNA, or the ribosome's job, is to read the mRNA, which is the message, and when it reads that message, it uses the instructions to make a protein. That protein is made from amino acids, so tRNA, or transfer RNA, is what's going to bring the amino acids to the ribosome. So here we have the whole process summarized in an image here. The nucleus of the cell, remember the nucleus contains the DNA. DNA is transcribed into mRNA, and here is our strand of mRNA coming out of the nucleus. So the mRNA is the instructions. The ribosome here is reading those instructions, and as it reads those instructions, it brings in the tRNA, carrying with it an amino acid, and uses those amino acids that are being brought in to ultimately make a protein. And you can see this chain of amino acids forming here at this ribosome and another one here at this ribosome at the end. Now, the amino acid sequence is what makes a protein the correct protein, and that is coded by the mRNA base sequences. Remember that this process is called translation. Well, what does translation mean in real life? It means you're taking a message in one language and you are converting it into a different language. So the language of the cell is going to be, first of all, the language of nucleic acids. mRNA is a nucleic acid. It's written in nucleotides, so that's one language. But the second language, what we're turning it into, is going to be the language of proteins. Proteins are made out of amino acids. So that's why we call this process translation. The cell, specifically the ribosome, is reading the language of nucleic acids, and then it is outputting a protein, which is a completely different language made out of amino acids. That mRNA, when we say it's being read, it's read 3 nucleotides at a time, and this is really important. It takes 3 nucleotides to form a code. If you remember from the POGL that we did, that triplet code, 3 nucleotides on mRNA is going to be a code for 1 amino acid. So those triplets of nucleotides, we call that a codon. Very important vocabulary term to understand. So if we look at this strand of mRNA here, we can see this first group of 3 bases. That would be the first codon, AUG. Now, if you remember from that genetic code that we reviewed in the POGL, AUG is the code for the amino acid methionine, or MET is the abbreviation for that amino acid. This next group of 3 here, that would be our second codon, CUU, and this codon is the code for leucine, or LEU. So let's look at that genetic code here. This is a similar version to what you saw in your POGL. And you'll notice that the genetic code lists every single amino acid. There are 20 different amino acids, and they are all represented in this genetic code. But the amino acids aren't the only thing here. We also have 3 stop codons. Okay, here you can see the stop codons. And then there is 1 start codon, which is also the code for the amino acid methionine. So this is our 1 start codon here. And if you have forgotten how to interpret this genetic code, we'll go through this quickly here. But if you need extra help with it, please reach out to me and ask me to help you out or give you some more practice interpreting genetic code. So we have our 3 steps listed. If we're looking for the codon CAU, that's the example we're going to use. Our first step is to find the first base C over here where it says first base. So here is the C. That means that the code we're looking for is somewhere in this row. All of the codons in this row have C as their first base. Our second step, look across the top. Our second base is A. So everything with a second base of A is in this column. So now we've narrowed it down to this one box right here. And finally, our third step is to look for the U, the third base. So here we see U is our third base. We find that on the right-hand side. And so the very first thing in this box is going to be CAU, and we can see that is the code for histidine, the amino acid histidine. So let's do another one as an example. If we are looking for the code GCC, okay, go ahead and pause the video, find GCC, and once you have your answer, you can unpause the video and see if you got it right. All right, let's find GCC. Our first base here is G. Our second base is C. So if we go down to this box and look for C, GCC is alanine, or ALA is the abbreviation. So hopefully you got that correct. And if you did not get that correct, again, please reach out to me and ask for a little extra help. This is really important for you to be able to interpret the genetic code. So now that we know how to read the genetic code, let's talk about how those amino acids are linked together to actually become a protein. First of all, amino acids are carried by tRNA. Remember, tRNA stands for transfer RNA, has an anticodon. That anticodon is going to be complementary to the codon on a strand of mRNA. It's really important not to mix up these two. You do need to remember that the anticodon is a part of a tRNA molecule, and the codon is on mRNA. One good way to remember this is think of the T in anticodon and the T in tRNA, and that will help you remember which one it's a part of. Now, the job of tRNA, the T stands for transfer, so it transfers or carries an amino acid on the end of it, and that amino acid is specific to the anticodon. Well, what does that mean? That means whatever these 3 bases are down here are going to determine what amino acid is being carried at the top. Okay, that will be really important because remember, the anticodon is going to be complementary or going to match up with an mRNA codon. So different mRNA codons are going to need to code for different amino acids. And it's by matching up with the anticodon that the correct amino acid gets added in the polypeptide chain. So now let's talk about the ribosome, because that's the cellular structure that is actually doing the construction of the protein. Ribosomes have 2 subunits. They're called the large and the small subunit. The large subunit is responsible for binding the tRNA. It has these different locations where tRNA molecules can come in and be held in place by this ribosome. And the small subunit binds to the mRNA. The mRNA would be found down here in the bottom of this diagram. And that's more responsible for holding the mRNA and holding it so that it's read in the proper position. So let's go through the steps of translation. Now keep in mind that this is an ongoing process. So what we're going to do is we're going to go through an overview of the steps, and then I'll show you a quick animation that will help put it all into context. I think it helps to see it all kind of moving together as one process. So our first step of translation here is going to be the tRNA binding to a start codon. We have to start the process, and it's the start codon that's the signal for where translation is going to begin. Our next step, after we have set our start location, the next thing that happens is a tRNA molecule is going to come in that is complementary to the next codon of the mRNA. So let's look at that in a diagram. Here was our first step of binding this ribosome to the start codon. So we have our first tRNA is attached to the start codon, the ribosome forms around it, and then in step 2, we have this tRNA coming in to that exposed slot right here. Now again, this codon is going to be complementary to this anticodon. So that's how we make sure that the correct tRNA is added. A tRNA that doesn't have the right anticodon is not going to fit here. It's like puzzle pieces. So this is how the ribosome makes sure that only the correct tRNA is added in next in the protein. So our third step, now that we have the amino acid in the ribosome, we're going to form a peptide bond to form that amino acid into the protein. And then once that is bonded, it's going to pull the mRNA strand through so that it can read the next codon. Okay, so here was our initial amino acid. We're bonding that leucine, that was the second amino acid that came in. So formation of that peptide bond, that was our step 3. And then not shown in this picture, the ribosome is going to pull that mRNA through. So now this codon, this is the one that is going to be exposed next. So it's going to move everything over by 3 bases so that we can have a new tRNA come in. Okay, so the empty tRNA is leaving. Here you can see we've slid that mRNA through, so a new one is coming in. So these 2 steps are kind of happening at the same time, right? Step 5 is talking about this empty tRNA leaving. Step 6 is talking about this new one coming in, but really they're both happening simultaneously. It's not like one happens first and one happens second. They both happen at the same time. So this ribosome is continuing to move down the strand of mRNA. Each new codon that's exposed is going to bring in a tRNA molecule, and then the one that has dropped off its amino acid is free to leave. And then finally, our last step here, this is just going to continue until we reach a stop codon. The stop codon, just like a start codon was the code to start making a protein, the stop codon lets the ribosome know when the protein is finished. When the stop codon is read, the whole thing will disassemble and that protein is now free to be used by the cell. So let's look at this in action here. I have this animation and we have a strand of mRNA here at the beginning. You can see the start codon here is going to be where that tRNA comes in, and then the ribosome will assemble around it. So we see that taking place. And then once that occurs, now we have an exposed site for the next tRNA to come in. Its anticodon is complementary to this codon. And then the process just continues. One tRNA after another comes in. Its anticodon is always complementary to the codon, and that happens until it gets to a stop codon. The stop codon is the cue for that protein and the ribosome to disassemble. So I want to leave you here with this one final summary image. We've now talked about all of the steps of the process. Hopefully all of these labels now make sense to you. We have the mRNA strand, which is the message that came from the nucleus. That's being read by the ribosome, this large teal structure here. And when it reads this group of 3 bases, each group of 3 bases on mRNA, we call that a codon. Each codon is complementary to an anticodon on the tRNA molecule. Remember, tRNA is bringing with it an amino acid to the ribosome. That's how the amino acids get there. And the correct amino acid is added because the anticodon only matches up with the complementary codon. So the tRNA with this amino acid always has this anticodon, which means it will only match up with this codon. And that's how we get the correct sequence of amino acids. The peptide bond is forming between amino acids to make this protein chain. And this process continues until we reach a stop codon on the strand of mRNA. When the ribosome gets to the stop codon, the process is finished, everything disassembles, and this amino acid chain is free to become a fully functional protein. So in closing, here you have a copy of the 8.5 section quiz from your textbook. Go ahead and look at these 5 short multiple choice questions. See if you can answer them and if they make sense to you. Keep track of your answers so that we can discuss them in class. And hopefully you found this to be helpful. As always, if you have any questions as you're studying, please feel free to reach out to me.